Hello everybody and welcome to our module on ELISA. ELISA stands for Enzyme-Linked Immunosorbent Assay, and it is a laboratory technique that can detect antigens and antibodies in serum. So in other words, this is a very good technique for detecting proteins found in our bloodstream. The way it works is via an enzymatic color change. We'll talk more about this in a minute, and there are several different variants of the ELISA test. One is called the direct, there's also the indirect, the sandwich, and the competitive. We'll talk about all of them in this video today. ELISA is often done in a specialized laboratory plate with a collection of wells where samples can be added for testing. This is an example of one shown on the screen here. We'll start by talking about the direct ELISA technique. In this test, you add the serum you want to test to one of those wells we saw in the last slide, and the serum will coat the plate, and the plate is designed so that antigens will stick to the surface. You then wash away the fluid. So this is a picture of what happens in one of those wells at the bottom of the screen. It's empty, you add serum and then wash away the liquid, and you are left with antigens bound to the plate. You then add an antibody specific to the antigen you wish to test for, but this antibody has been modified and it has an enzyme attached to it. This is called an enzyme-labeled antibody. This is shown at the bottom of the screen, so our antigen has been adhered to the surface of the well. We then add the antibody to that antigen, and that antibody is attached to an enzyme shown by the letter E here. You then wash away any unbound antibodies, so the antibodies that are enzyme labeled will only remain behind if the antigen you are testing for is present and stuck to the plate. You then add a substrate for that enzyme, and the enzyme converts the substrate and creates a color change that you can see with your eye. So if you see a color change like the yellow I've shown here, it means that the antigen was present in your initial test sample and therefore it stuck to the surface, and then the antibody bound to that antigen and the enzyme converted the substrate and created the color change. And the reason this is called a direct ELISA test is because the enzyme-linked antibody directly binds to the antigen. A variant of this is called the indirect ELISA. In this test, you add the serum for analysis to one of the wells, just like in direct. I've showed that at the bottom left side of the screen here. You then add an antibody to the antigen of interest, once again as shown here. However, in this situation, the antibody is not enzyme linked. You then wash away any unbound antibody, so antibody will only remain behind if the antigen was present in your initial test sample. You then add an enzyme-labeled secondary antibody that binds to the first antibody that is bound to the antigen. So this gets confusing. You've got multiple antibodies here binding to one another. You then add the substrate, and that will create a color change if the antigen is present and has bound to the first antibody, which has then bound the second antibody. So the net result is the same as the direct ELISA. It identifies the presence of an antigen in the serum when you see the color change. What's different from the direct ELISA is that the enzyme-linked antibodies indirectly bind antigen. They do so via another antibody. So as you can see, the direct ELISA is simpler, and you might wonder, why not just always use the direct ELISA? Well, let's talk about that. The direct ELISA has fewer steps. However, the specific antibody has to be enzyme linked, and it's very time consuming to label antibodies to unique antigens. In other words, for every antigen you want to test for, you must have an antibody to that antigen which has been linked to an enzyme. The indirect ELISA, in contrast, has more steps, but the specific antibody is not enzyme linked, and this means that specific antibodies to antigens are easier to acquire. For example, you can inject a mouse with an antigen and quickly obtain antibodies to that antigen. They will not be enzyme linked, but you will get the antibodies very quickly. You can then have hanging around in your lab lots of secondary antibody that's very easy to acquire, that is just a generic anti-mouse IgG antibody, and this makes it very easy to do the ELISA on lots and lots of different antigens. And so for this reason, because it's easier to acquire the antibodies for the indirect ELISA, this is often done more commonly. It's time consuming and difficult to label all the different antibodies to unique antigens for the direct ELISA. Another variant of ELISA testing is called the sandwich ELISA. In this type of testing, the plates are coated with an antibody called a capture antibody. The sample you want to test is then added, and if antigen is present, it will bind to the capture antibody. I've shown this at the bottom of the screen here. We've got a capture antibody at the bottom, and it is binding the antigen you are testing for. You then add a detecting antibody which also binds to the antigen. This is why it's called a sandwich, because the antigen will be sandwiched between two different antibodies. You can use either the direct or indirect ELISA technique then to identify if antigen has been bound. If you want to do this with the direct technique, the detecting antibody will be enzyme linked. If you want to do this with the indirect technique, then a secondary enzyme linked antibody must be added. In this example at the bottom of the screen, I've shown a secondary antibody here that binds to the detecting antibody. This would be an example of the indirect technique. You then add substrate, and if the color changes, it means that that antigen was present in the sample you tested. The advantages of the sandwich ELISA are that it has a high specificity. Because two antibodies are used, it's very unlikely to bind the wrong antigen. It can also be used with complex samples. The antigen does not require purification, and again, this is because two antibodies are used, so it's very unlikely to bind the incorrect antigen. And then finally, because it can be used with a secondary antibody like the indirect, it has all the advantages of the indirect, making it easier to obtain the enzyme-linked antibodies. The last variant of ELISA we'll discuss is called the competitive ELISA, and this type of ELISA test is very different from the others we've discussed up until now in this video. In a competitive ELISA test, a primary antibody is incubated with a sample, and if antigens are present in the sample that bind the antibody, then antigen-antibody complexes will form. The key principle of a competitive ELISA that you have to appreciate is that the more antigen that is present in the sample, the more binding there will be to those primary antibodies and the less free antibody that will be left behind. To appreciate this, let's look at the drawing I've made on the screen here. We've got 3 molecules of antigen and they are binding to 3 antibodies. This is leaving 3 other antibodies behind that are free or unbound. If there had been more antigen present in this sample, there would have been less free antibody left behind, and that'll be important when we talk about how to interpret this test. So after you mix the sample with the antibodies, the mixture is added to antigen-coated plates. Any unbound antibody, that's the free antibody we saw in the last slide, will bind to the antigen on the plates. You then wash away antigen-antibody complexes and a secondary antibody and substrate are added. And the key difference here between this type of ELISA and the others we've discussed in this video is that if there is more color change, this means there was less antigen present in the initial sample. This will make more sense once we go through this picture on the screen. So in the sample at the top left of the screen, there's lots of antigen present as indicated by the 5 red diamonds. And those 5 red diamonds representing antigen are all binding to antibodies. This is leaving only one single lone antibody behind that is free or unbound. We then add the mixture to plates coated with antigen, and that one free antibody binds to the antigen on the plates. The other antibody-antigen complexes are left in solution and they will be washed away. We then add our enzyme and our substrate, and what we get is just a little bit of color because only that one free antibody bound to the antigen on the plate. So in this type of ELISA testing, less color change meant lots of antigen present in the initial sample. At the bottom of the screen, I've shown an example where we have very little antigen. We've just got one red diamond here, so it is binding to one single antibody. We then add the mixture to the plates, and all the free antibodies bind to the antigen on the plate, and that one little antigen-antibody complex is washed away. Now when we add our enzyme and substrate, we get lots of color change because we had all that free antibody initially that did not have any antigen to bind to. So in this case on the bottom, more color change meant less antigen in the initial sample, and that's how a competitive ELISA test works. One of the major clinical uses of ELISA testing is in diagnosis of HIV infection. You can use ELISA testing to identify HIV antibodies in the serum. The indirect method is often used, although there are many variants in commercially available kits. In many of these kits, the HIV antigen is attached to wells. You can then add your sample to the wells and it will react with the antigen-coated plate. If there are HIV antibodies in the sample, it will bind to the HIV antigens in the plate. You then add a secondary antibody, which is anti-human immunoglobulin with bound enzyme, and you add a substrate, and if there's color change, it means that there is HIV antibody present in the serum you tested. You can also use ELISA testing to identify the HIV P24 antigen in serum. This is often done with the sandwich ELISA, although once again many variants are used. And in the modern era, HIV diagnosis is often done with combination tests that look for both antibodies and the P24 antigen. And that concludes our video on ELISA.